So good afternoon. I'd like to extend a very warm welcome to today's webinar from the Physiological Society. My name is Morag Mansley. I'm from the University of St Andrews. My co-chair is Mike Althaus from Bonn-Rhein-Sieg, University of Applied Sciences in Germany. Together we are the theme leads for the epithelia and membrane transport theme within the Society, and it's our great pleasure to host today's webinar, The Lung Epithelium, the First Line of Defense Against Respiratory Viruses. We thought it was a particularly apt time to hear from scientists working in this field, and they're going to discuss their research on how the epithelium interacts and responds to respiratory viruses. So just as a brief overview of today's session, each of our speakers will deliver a 20-minute presentation back to back, and then we will have a 15-minute question and answer session at the end. So please submit your questions using the Q&A button. You can do this throughout the talks, but Mike and I will moderate these at the end in the 15-minute session. If you would like to include your institution, then we can say where you're from, which is quite nice. But you can also submit your questions anonymously if you would prefer. We will try to get through as many questions as possible, and I'm sure there will be a lot to discuss. As ever, we welcome all questions, and we would encourage our early career members and students to join in. And finally, please note that this session is being recorded and will be available to watch later. So I'm going to pass over to Mike quickly.
Okay, thank you very much, Morag, for this lovely introduction, and a warm welcome and greetings from Germany. It is my great pleasure to introduce our two speakers for today, Dr. Claire Smith from University College in London and Dr. Oliver Wittekind from Ulm University in Germany. And they are both experts in lung epithelial biology. And as Morag said, we thought it is very timely to talk about how the lung epithelium acts as a first line of defense against respiratory viruses. The first speaker will be Claire Smith, and she will be talking about a window to the lung, observing the lung epithelial interface during viral infections. And with this, Claire, I'd like to hand over to you.
All right, I'd like to take this opportunity to thank Mike and Morag for giving me this opportunity to speak, and for all of you who are joining today. Yeah, I hope you enjoy the presentation. My group is based within the Infection Immunity Inflammation Department at UCL Great Ormond Street Institute of Child Health. Our research focuses on how the airway protects us from infection. And in doing so, we've developed several human infection models to help us unravel clues about what happens during the very early stages of viral infection. I've titled this talk A Window on the Lung because most of our research uses visual techniques. I think a picture paints 1,000 words, and we believe that we can get much more data and deeper understanding from observing what happens during infection. Today, I'm going to talk about how the initial interaction of the virus, the lung epithelium, and also immune cells can drive disease severity, and how we've applied our techniques to tackle the current COVID-19 pandemic. Now, much of what I tell you today will come from data collected looking through a very small window, less than 1 centimetre in diameter. But it's important to consider that even small changes can result in a ripple effect that can have dramatic downstream effects, not only in the lung, but also throughout the body. So the SARS-CoV-2 coronavirus that causes COVID-19 disease is 50 to 200 nanometers wide. To put that in context, a human hair is approximately 80 to 100,000 nanometers wide. And that means that SARS-CoV-2 is 500 times smaller than the width of a human hair. Now, we know that viruses are transmissible in the air, and the image on the right shows the effectiveness of a surgical mask containing these particles released from a sneeze. I wanted to find some papers about how many particles were released when we sneeze, but in the not so extensive search that I did, the closest data I found was that from patients with influenza, they're shown to release up to 1 million particles per cough into the environment. So that just goes to show just how many of these particles are existing around us and how easily they are inhaled. So why, with so many particles and viruses circulating, do we not get sick all the time? Well, this is because our airways have developed many ways to prevent infection. These include physical barriers, such as small openings in the nostrils and the pharynx, and hairs that trap many organisms and allergens before they're able to reach the more vulnerable tissues. And this image just shows a nice electron micrograph of a pollen grain stuck to a nose hair. If we look closely at this airway epithelium under electron microscope, we can see that it also is covered in microscopic tiny hairs, which are motile, called cilia. The cells that line the airway each possess up to 250 of these motile cilia, which beat in a synchronous wave to propel pathogens and mucus and trapped debris away from vulnerable cells. Our research has focused on studying the role that these cilia play in preventing infection, and also how they can be the target of many infectious agents. To do this, we take a sample of the airway by brushing the epithelium with a very small brush. This removes fine strips of the surface cells and leaves submucosal layer intact so that it can regrow. These samples are then placed directly onto a microscope slide, and using slow motion video microscopy, we can view the beating cilia. These cilia beat 16 times a second. So in this video, we've slowed it down considerably so that we can see the beat pattern with the naked eye. And this video nicely demonstrates how these cilia are beating in a synchronous way and helping to clear debris around this epithelial layer. So we can see some dead cells moving across in the direction that the cilia are beating. Oh, sorry. Another way that our bodies have learned to fight infection is through the rapid recruitment of immune cells. This video shows circulating white blood cells known as neutrophils, which are attracted to the site of infection by the release of chemokines by the damaged tissue, and they're the green dots that are shown here. Neutrophils are able to move from the bloodstream into the tissue by following this signal along a concentration gradient towards the site of an infection or where there's cell damage. This particular video shows some bacterial infecting. Once they get there, the neutrophils can phagocytose or consume the pathogen, and in a controlled manner, they can destroy it. Methods have been developed in order to study this type of immune cell recruitment in animals. The method shown here is known as intravital microscopy, which is what's used to observe what happens when the tissue under the ear of a mouse is damaged. This video shows neutrophils stained with a red dye and macrophages stained with a green dye. At the center here, we have an area which has been damaged by a laser pointer. We can see that, and this is the time, that the neutrophils are rapidly recruited to an area of damage, and this is followed by a slower but more purposeful migration of the neutrophils. Using this technique, we can track the movement of these immune cells and study their coordination. This can help us understand more about the role that these cells play in the fight against early infection and how, in some cases, this can go wrong. In order to study this in humans, we've developed a very different kind of model, but using the samples that we're able to get from the airway epithelial cell brushings that I showed you earlier, we can expand the progenitor cells in cell culture. So these are the cells which would go on to make the fully differentiated epithelial cells that I showed you in the electron micrograph. So the progenitor cells are seeded onto a porous membrane, which allows us to feed the cells from just in one direction. So we create an air-liquid interface, much like you would find in the lung. This exposure to the air promotes the cells to differentiate into the different types of cells that we see in the airway. So we have mucus-producing cells and ciliated cells, and these ciliated cells have motile cilia as well. So it really is giving us a small sample of what the lung, of the original lung or air nasal sample that we get from the donor. And once we have these cells in culture, we can infect them with different viruses to study how they respond to infection, how much virus they're replicating, and how much inflammatory response that they have. Now, I'm just going to change direction a little bit to tell you about the virus that I've been working on for many years, known as RSV. And this is a very common virus that actually very few people have heard of. It infects up to 70% of all infants in their first year of life. So it's very likely that you've experienced this virus within your lifetime. 3% of these infants that are infected will require hospitalization, but we don't really know why some infants are getting more severe disease while most actually have very mild disease, as 80% of these infants have no underlying health conditions. But very recent data has, from sampling infants with viral bronchiolitis suggests that, curiously, severe RSV disease is associated with a lower virus concentration in the lung and lower interferon, which is not what we would necessarily have expected to see. So this is very interesting development. But using our model, we found that this virus is specifically targeting these ciliated cells for infection. In this image, we can see that we can see a sort of side view of these epithelial cells. So here we have cilia with an uninfected epithelial cell. So we just see the cilia stained in red. In this example, which is 20 to 24 hours after we infected them with RSV, we can see the cilia still in red, but we have the surface of the cell expressing viral protein, which is shown in green. After that time point, so after a 24-hour period, moving up to 72 hours post-infection, we're also seeing viral antigen displayed on the surface of the cilia. So we've got viral antigen on the cell and also in the cilia itself. And this is specifically, say, we found that this virus is targeting the ciliated cells. And other people have shown similar data. This is another group who've done similar work, showing that the virus is targeting the cells. So there's cells here as well in these black areas, but there's no virus infecting those cells. So the virus is shown in green. So the virus is targeting these cells that have motile cilia. We don't know why, but it's possible that this could have a very dramatic effect on mucociliary clearance. And this just shows you some of the other data that we got from our cultures, which was quite interesting, because we find actually that this virus isn't producing a lot of cytopathic effects. So although we're getting quite a lot of viral replication and we can see lots of virally infected cells, the cells don't appear to be very damaged in our model. So this image shows a nice healthy epithelium. So we have a nice straight edge and full length cilia being shown. And this is a transmission electron micrograph. So this is where we cut through the cells so we can see them side on. In this image, we can see that this cell is starting to move away from the epithelium. So we don't have this nice straight edge anymore. We're starting to get some cells breaking away, and these could eventually move off. In this image, we can see some ciliary loss. So we don't have as many cilia as we see in A, in our nice healthy epithelium. And in B, we see some cytoplasmic blebbing. But again, other than that, we're not seeing dramatic tissue damage, which we would expect to see as we see that in children with RSV. But one thing that we do see, which seems to be consistent and was quite a new observation, is quite an increase in ciliary dyskinesia. So as you remember in that very first video that I showed you with the cilia beating side on, those cilia were all beating in a very synchronous way. Whereas with RSV infection, we're starting to see these cilia aren't beating in a synchronous way. And these ones, these particular cells here are beating against each other, which means that quite early on in infection, we might be seeing an effect on mucociliary clearance. And this is something that you probably all experienced at some point in your lives, where you have quite an early infection. You might not feel particularly ill, but you start to get a runny nose. And this is the type of, this can be the cause of that runny nose, because if you haven't got cilia effectively clearing any mucus that you have in your airway, then it's just going to come out the other side. So we were quite curious about why our model wasn't necessarily completely representing what we were seeing in the clinical situation. And so we went back to start looking at what's at the lungs of infants with RSV. And one thing that's very distinctive about these cases is that their lungs are full of neutrophils. So let's say this original immune cell that infiltrates the airway in response to infection, well, they don't seem to be getting rid of them in these children. So we've been detecting these large plugs blocking the airways, which are full of neutrophils. So this led us to think, well, maybe we should be incorporating neutrophils into our infection model. And so we developed, and this has just been in the last 2 to 3 years, we've developed a new model where, again, we take our airway epithelial cells from our healthy donors, we expand them in culture just like we did before, but rather than seeding them onto the top of these porous membranes, we now seed them onto the bottom. And there's still an air-liquid interface, but they're grown upside down. And the reason for doing this is that once we've infected the epithelial cells, we can take some blood from another donor, or in some cases the same donor that gave us the airway epithelial cells. We stain them with a fluorescent marker, and we put them on what would be the bloodstream side of the epithelial cell. So we can, and this animation kind of demonstrates what we do. So we stain our neutrophils, put them onto the bloodstream. Sorry, put them. Oh, not working. Sorry. We put them onto the bloodstream side, and then there should be an animation, but it's not working, so I apologize. And then we can watch as they migrate across the epithelial cells and drop into what would be the airway surface fluid. And we can place these cells onto a microscope, and with time-lapse microscopy, very fast time-lapse microscopy, we're able to track the movement of these neutrophils. And this video here just shows one of the videos that we've been able to capture. We've got our red stained neutrophils. The black dots that you can see are those pores in the membrane, and the green staining is the viral infected epithelial cells. So you can see that very rapidly we're getting neutrophils squeezing themselves through these pores, and they're moving around the epithelium, and they seem to be collecting themselves in particular areas. And we can track the movement of these neutrophils, much like we saw in the mouse model. We can, because this is a time lapse, we can trace how each individual neutrophil moves across in this model. And what we found, and this is quite preliminary data that's been conducted by Lizzie Robinson, where we found that when we expose neutrophils to an epithelium which isn't infected with anything, or mock-infected epithelium, so our control, we find that the neutrophils move in a much more vertical direction. So they're coming through the pores, across the epithelium, and then they're dissociating into the airway surface fluid. But in our RSV-infected cultures, the neutrophils are coming through and they're moving across in a much more horizontal direction, implying that they could be rolling across the epithelial layer. And this is what we find when we analyze this in more detail. So after an hour of exposure to the neutrophils, if we stain the epithelium and look in more detail about where these neutrophils are associating, we can find that in the mock-infected epithelium, we have a lot more sort of uniform collection of neutrophils. So there doesn't seem to be any real association to particular areas. So here neutrophils are pink and the epithelial cell nuclei is in blue. But in our RSV-infected epithelial cells, we get a lot more neutrophils adherent to the epithelial layer, but also we can see that they're starting to form these quite large clusters. And this is something that we've published earlier this year, but we're very intrigued about how these cells are forming these clusters and what implications these clusters are having on disease severity. One thing we do know is that this influx of neutrophils into the airway is causing a reduction in the amount of infectious virus. So their movement into the airway is a positive thing in one respect. Say after 1 hour, we start to see a slight reduction. Here we can see how much virus we have in the cultures that doesn't have the neutrophils, and then how much we have once an hour after we've added the neutrophils. And this becomes significantly different after 4 hours. And just to demonstrate this, we found this very nice video of this particular epithelial cell, what happens to it once these neutrophils come through the membrane. If you watch closely, you see after about 40 minutes, that cell disappears. So it's like it's been phagocytosed or eliminated by this influx of neutrophils. Now, of course, nothing is ever straightforward in immunology or in fact, in a lot of biology. And unfortunately, the influx of neutrophils also leads to some collateral damage of the airway tissue. So where I was showing you before, where we don't actually see a lot of cytopathology in our electron microscopy images, here, once the neutrophils are coming in, we see a lot of cytopathology. So this video I'm going to show you now shows a much longer time course. So we're going over a 24-hour period. The neutrophils, again, are stained in red, and the epithelial epithelial cells, the RSV infected epithelial cells, sorry, are stained in green, and the rest of the grey image is the bright field, so that's other cells that are in the image. But you can see that quite rapidly once the neutrophils start to apoptose or die off, they've lost their red stain, we start to see that this epithelium starts to pull away and round up. And if eventually, if that was in vivo, we would expect to see it be lost because this is a static culture, it's slightly different. But yeah, we start to get, excuse me, how do I get out this one? So yeah, so we're getting a lot more cytopathology now in our model, and this is leading to a reduction in ciliary beat frequency. So here we've got our mock infected epithelial cells that are with the neutrophils, and then when we look at our ciliary beat frequency with our RSV infected epithelium with neutrophils, we see that again at this point when these neutrophils start to die off at about 4 hours, we start to get a lot more a reduction in ciliary beat frequency. So our cilia aren't beating as fast, they won't be as effective as at clearing the virus and any debris, and they're significantly slowing down. And this seems to correlate with an increase in neutrophil elastase and other harmful proteins which are released by neutrophils. So this graph just shows that on the apical side, so that's the airway side, we're getting a lot more of this neutrophil elastase being released than we see from the neutrophils which are on the bloodstream side. So once these neutrophils are coming through the epithelium, they're changing their phenotype, they're becoming, well, some of them anyway, are becoming quite destructive. They're releasing harmful proteins into the supernatant, and this could be contributing to the reduction in viral load, but it's having some collateral damage effects as well. So given these two differential response, what we think is happening is that actually we've got two different types of neutrophil in this environment. So we think that one type is having a very controlled killing of the virus. It's phagocytosing any virus or viral infected cells and using this controlled method that was shown in the video earlier to apoptose and get rid of the virus. But then we think we have this other subtype of neutrophil, which is more towards a degranulation or a release of these harmful proteases into the airway supernatant. And so what we think is critical is to understand why we have one subtype or the other, and whether this kind of balance between these two types of neutrophils could play a role in the development of severe disease. And this is particularly relevant to the COVID-19 pandemic because neutrophils and their behavior has also been implicated in disease severity already with this infection. And the reason I kind of now draw into the SARS-CoV-2 is because there are actually a lot of similarities between the virus which causes COVID-19 and RSV that we've worked on for many years. So both of these viruses have been shown to infect the ciliated airway epithelial cells. Both have a differential response due to age. Whereas in RSV we see disease severity in the very young, in adults we have very mild disease, whereas in COVID-19 we see that it's much more severe in adults and the elderly compared to infants. And this is the focus of our current work, which has just been funded by the BBSRC. In this, we're going to use the two airway models that I've described today to study how the airway epithelium in children, adults, and the elderly differs in its response to SARS-CoV-2 infection. As we can see from the data, we get a lot more severity after the age of 50. That's when our case fatality ratio increases. And so we're particularly interested to see how the airway in the older generation is responding to the virus compared to that from children. So just to conclude, from our work, we're saying that it's very important to study this initiation of infection and host defense because we think that it could be leading to an inflammatory cascade. We're particularly interested in characterizing neutrophil subtypes in our airway model to see if we can help to predict the development of severe disease. And the other thing we're very focused on is using human models to study human viruses. This will provide relevant understanding and also help us develop a very relevant preclinical model that can be used for drug discovery. And I just want to finish up by thanking all the people that helped bring this work together, including Roz Smith, Lizzie Robinson, Jenny Herbert, Yu Dang, and Lou Ren, whose work I've presented here. And I also want to thank the people who've helped keep me sane over the last few months, which has been a real challenge during the lockdown. That's Rob Hines, Sam Ellis, Daniela Cardinale, and Danny Lee. And thank you for listening. Oh, and one other thing, if you're interested in joining our group as a postdoc or research assistant, I have 2 job vacancies that will be advertised shortly, so please get in touch. Thank you.
Thank you very much, Claire, for this fantastic talk. I was really amazed to see all these cells moving and squeezing themselves through the epithelium. Before we continue with our next speaker, I just want to mention again to the people who joined us a bit later, if you have any questions to Claire, please use the Q&A button at the bottom of your screen, and Morag and I, we will read out these questions at the end of the session. And with this, I'd like to hand over to Oliver Wittekind, who's from the University of Ulm in Germany. And Oliver's talk is about the lung epithelium during disease conditions. Oliver?
Yeah. I hope you can see my slides. Just a moment. Yeah. Thank you for this kind introduction. And it's a pleasure for me to give this talk here in this video chat. So I want to talk about the lung epithelium during disease conditions. Well, the airways, or the conductive airways, the respiratory airways, and the alveoli, they constitute a so-called air-filled compartment. And this air-filled compartment is separated from the organism by the lung epithelium. So this lung epithelium exposes its apical surface always towards the air-filled compartment, and therefore this surface has to be shielded by a liquid layer. And this liquid layer is produced by the epithelium itself. The epithelium secretes water on top of its apical surface, and so into the airways, and it balances the amount of water simply by adjusting the water resorption. So this is depending on its barrier function, and it's depending on its transport function. The liquid layer on top of the conducting airways is called the airway surface liquid. It has a height ranging between 20 to 150 micrometers, and this liquid layer established the so-called air-liquid interface. It has 2 different layers there. There's a mucus layer and a periciliar layer. The mucus layer is enriched by mucins, and therefore it's high viscose. That makes it a beautiful trap for inhaled particles and pathogens. Those pathogens can easily be trapped in this mucus layer, and they can be immobilized there. The periciliar layer has a low viscosity, and that enables the motile cilia to beat. And because of this concerted beating of this motile cilia, there is an ongoing transport of this mucus layer upwards towards the most proximal part of the airways in order to remove this mucus layer from the airways, and therefore also the trapped particles together with this mucus layer. So this is the clearance function of this epithelial cell layer. Well, in lung disease, we are facing a quite complex disease situation. There are regions in the lung that remained almost unaffected by this disease conditions, and others, they are heavily affected. In this affected region, we usually observe so-called inflammatory conditions. That means we have elevated levels of different cytokines that are released by the epithelial cells themselves, and cytokines that are released by the immune cells that are recruited at the site of damage. We have elevated ASL volume levels. That means we are dealing with an impaired barrier function and an impaired transport function of this epithelial cell layer. And of course, we are often observing reduced partial pressures of oxygen in these areas. So all these conditions, they contribute to the epithelial damage and they contribute to the lung damage, but they also triggers the activation of mechanisms, especially of mechanisms inside the epithelial cell layer that compensates for this damage and that restores or maintain almost homeostatic conditions even after upon those damaging situations. So when someone overlays the epithelium with an additional volume of, for instance, isotonic solution, it is easily to observe that this volume becomes resolved over time. So this volume is simply removed from the apical surface by water resorption. And when we performed those experiments, it turned out that there are two resorptive states in the epithelial cell layer. There is a low resorptive state and there is a high resorptive state. The low resorptive state depends on an ENaC activation, and this ENaC activation happens within the first couple of minutes after apical volume loading. ENaC is the ion channel that limits the water resorption simply by limiting the transcellular sodium resorption that drives the water resorption. And this channel becomes activated, and the activation reaches its maximum within 10 to 15 minutes. So this is a rapid effect. And we believe that this effect contributes to a short-term stabilization or balancing of this ASL volume level. The high resorptive state, the onset of this high resorptive state happens couple of hours later. So we observe the onset within 4 to 5 hours after apical volume loading. And there is a massive increase of this water resorption. And of course, this water resorption still depends on ENaC activity. However, there's something else that is going on because its onset happens even after the ENaC activation is already completed. So, and the something that is going on is the upregulation of an aquaporine, namely aquaporine 3. And this aquaporine 3 accumulates in the lateral membranes of these epithelial cells. And because of this accumulation, there is an increase in lateral water permeability of this epithelium. And we came up with a model that predicts that sodium, during the low resorptive state, sodium is taking up across the apical membrane through these ENaC channels. Then sodium is pumped by sodium-potassium ATPases into the lateral intercellular space, from where it can leak back through a diffusion barrier on top of the apical surface. So there's a recirculation of sodium always going on, and this recirculating sodium does not drive the water resorption. The water resorption that is driven by the sodium depends on the sodium that is guided from the lateral intercellular space into the basal compartment. And only this proportion of sodium that is guided into the basal compartment drives the water resorption. Upon an increase in lateral water permeability, we observe there is an increase in that sodium proportion that ends up here in the basal compartment. And that's the reason why we observe the increase in water resorption. So this model tells us two things. First of all, it tells us something about the importance of aquaporins in compensating, in compensatory water resorption that compensates for edema situations. It tells us also something about the diffusion barrier, because if the diffusion barrier would inhibit the back leak of this sodium, it would increase the water resorption. This diffusion barrier is built up by tight junctions, and this tight junction depends on the claudins. Claudins, they are transmembrane proteins, and these transmembrane proteins can interact with claudins of one and the same epithelial cell. And because of this interaction, there is the establishment of those multiple parallel strands that surround the cells at the apicolateral side. These strands are interconnected with other claudins, and these claudins of these strands can also trans-interact with claudins of the neighboring cell. And because of this trans-interaction, there is the pore formation into the tight junctions. And these pores that are formed by the claudins, they have a certain perm cell activity. That means they favorize the permeability of positively, negatively, or uncharged molecules or water. The other mechanism that contributes to the tight junction permeability depends on reversible and dynamic strand breakage of these parallel strands. And this pathway is utilized by larger molecules for permeating the tight junctions, for instance, like proteins. So both pathways contributes to the paracellular permeability, and they depend on the composition, on the clouding composition of these tight junctions. Well, when someone cultivates a lung epithelium at a liquid interface conditions, he or she would observe that there is an increase in the transepithelial electrical resistance. And the transepithelial electrical resistance is a measure for the ion permeability of these tight junctions. So an increase in the resistance means that there is a reduced ion permeability. So at a liquid interface condition there, we observe this establishment of such a resistance. That means there is the formation of tight junctions going on. And that is something what we also observe at edema conditions. So at edema conditions, there is always an establishment or a formation of these tight junctions. However, the electrical resistance never reaches the same values as it did during a liquid interface conditions. And this is due to the fact that there is a certain claudine, namely claudine 1, that is downregulated at edema conditions. And because of the absence of this claudine 1 in the tight junction, we observe the increase in paracellular ion permeability, and this can be measured simply by a reduced transepithelial electrical resistance. Well, claudine 1 is modulated or is controlled by retinoic acid signaling. At edema conditions, we observe that there is an increase in retinoic acid synthesis in these cells. The retinoic acid binds to its receptor, and upon this receptor binding, this receptor ligand complex inhibits the formation of the AP1 element. The AP1 element is a positive transcription factor that drives the expression of claudine 1, and because of the lack of AP1 element, we observe the reduction of claudine 1 and the opening of the tight junction. So the other thing that happens is that there are quite damaging effects due to the inflammatory conditions. Inflammatory conditions mean that there are elevated cytokine levels in this region of the lungs which are affected by the disease. These cytokines can be released by the epithelial cells themselves. For instance, upon a trigger that invading particles or pathogens interact with these cells, and these cytokines can attract immune cells to the site of damage. So there are cytokines released by the epithelial cells, and there are cytokines released by the recruited immune cells. And from all these cytokines, it turned out that only IL-13 and TGF-beta1, these two cytokines were the only cytokines that reduced the transepithelial electrical resistance in all epithelial cell layers from all investigated donors. So IL-13 acts on the tight junction simply by internalizing or triggering the internalization of all tight junction proteins. So in IL-13 treated cells, we observe the internalization of all claudines, of all occludines, as well as of sonolar occludine. And these proteins, they form intracellular granules. Other lateral contacts remained almost unaffected by IL-13. And IL-13 acts on the tight junctions via activating its canonical STAT6 pathway. This activation triggers the upregulation of an ubiquitin E2 conjugating enzyme, namely UBE2Z. And UBE2Z initiates then the polyubiquitination and subsequently internalization of these tight junction proteins. And because of this massive ubiquitination of these proteins, we observe an overflow of the proteasome, and this overflow triggers the damaging effect on this cell layer. So this results in the complete disruption of these tight junctions. However, the integrity of the cell layer per se remains intact because the other lateral junctions remained almost unaffected by this pathway. So there is a massive opening of the tight junction. That means there is a massive leak through the tight junction from sodium that recirculates and other solutes that enters the apical surface from the basolateral compartment. And therefore someone would expect that there must be an elevated or an elevation of the ASL volume level. However, this is something what we never observed in our experiments. So IL-13 did not affect the ASL volume level. What we observed instead was that there is an upregulation of an additional transport mechanism which works on top. So it works additive to the transport that is mediated by INEC germs. And this transport mechanism is amiloride insensitive. And we believe that this transport counteracts the leak through of solutes and water across this epithelial cell layer. However, unfortunately, we don't know which transport molecules are involved yet. This is an ongoing project, but probably next year, who knows. The other cytokine was TGF-beta. TGF-beta 1 acts via its receptors R1 and R2, and both receptors are expressed along the airways. Well, they are expressed only in the ciliated cells, and there they localize along the multiciliar. Non-ciliated cells remained almost negative for these receptors and these immune histochemical stainings. And this is something that is similar to our in vitro cell culture models of the airway epithelium that is based on primary cultivated bronchial epithelial cells. Claire gave you a beautiful introduction in this model already. So in this model, the two receptors R1 and R2, they both localized along the multiciliar. And upon TGF-beta 1 treatment, the R2 receptor became internalized into the cells, while the R1 receptor remained at the multiciliar. That tells us that TGF-beta most likely acts via the heteromeric R1, R2 receptors, and it triggers the opening of the tight junction by interfering with the localization of a specific claudin, namely claudin 3. So in the presence of TGF-beta, claudin 3 becomes internalized into the cells, and then it pops up in the nuclei. So TGF-beta shifts the claudin 3 from the tight junctions in the nuclei, and this is the reason why we observe the leakiness of the tight junction in TGF-beta treated epithelia. Well, we presume a model that these receptors that are located along the multiciliar, they can interact or they can bind to their ligand. And upon this ligand binding, these receptors, these activated receptors now travels along the multiciliar to the basis of the multiciliar where the phosphorylation of SMAD2/3 occurs. And this is a phosphorylation that is ALK5 dependent. So that's the kinase activity localized to these receptor heteromeres. And the phosphorylated SMAD2/3 complexes are transported into the nuclei, and they also interfere with the tight junctions. And this triggers finally the internalization of claudin 3 into the cell nuclei. Well, this is, I think, a quite an interesting model because it, first of all, of course, it tells you something how TGF-beta may interfere with the tight junction, but it highlights a role for this multiciliar as a sensing structure for larger ligands, because those larger ligands like TGF-beta, but also probably like other viruses like RSV and corona, those particles should be immobilized somehow here in the mucus layer, and they are hindered to get access to the cell surfaces. And if the cells want, or if the cell should with it is necessary to expose these receptors at the interface of these between the mucous layer and the periciliar layer. And this is achieved by localizing these receptors along the motile cilia. At the end of my talk, I want to thank all my colleagues that contributed to these projects, namely Hannah Schmidt, Carol Schilp, and Robin Lochbaum. They did a beautiful job on all these projects. Hannah Schmidt was working on the water resorption and the IL-13 project. Caroline Schilp elaborated the TGF-beta project, and Robin Lochbaum was focusing on the effect of elevated ASL levels on the tight junctions. I want also to emphasize Mike Kowal from Emory. We got a quite productive collaboration with him, and of course Peter Braubach from the Medical School Hanover. He supported our projects with lung tissues. Thank you for your attention.
Thank you very much, Oliver. That was a really excellent talk, and I'd like to thank both speakers for sticking to time, because that gives us a bit more time for questions. I don't know if Claire's still there. Hi there. So we've had a few questions in, and I'm going to get started with one from Jane Cleel, sorry. I think this is perhaps to Claire to begin with. Does RSV affect the cilia in other parts of the body, for example, the reproductive tract, and therefore could it influence pregnancy outcome?
That's interesting. I don't know of many studies that have been looking at RSV affecting cilia in other organs. I think there might be some work that's looked at it in the maybe in the gastrointestinal tract. I mean, the main thing is how the virus gets to these places. You know, there would be evidence to suggest that the virus could be in the gastrointestinal tract. I don't know anything about being in the reproductive tract. So I don't know. I don't necessarily, I wouldn't have thought so, but then, you know, you don't know until you've looked. So I don't know of anything like that.
All right. We're just going to alternate a bit between the questions so that you're used to Morag and me swapping over here. We have a question from Mirza Subhan from the University of Plymouth, who's thanking both speakers for the interesting talks. And a question again to Claire, is ciliary dyskinesia a protective mechanism during viral infection? And could it be artificially activated to help prevent COVID, or by the side effect would be a runny nose?
I don't think it would be a protective mechanism, because it is something which is quite innately important for clearing pathogens away. So any kind of ciliary dyskinesia, or even a reduction in ciliary beat frequency, would just mean that those viruses are hanging around a lot longer. I mean, if you think you've got this sort of periciliary layer, so you've got a mucus layer on top of your cilia, you want those to keep moving as quick as possible so that the virus can't sort of drop down to infect the cells. So you, in a way, you want your cilia beating as fast as possible to propel those viruses away from any, you know, not being able to get near to those, you know, receptors that they can bind to start the infection. So I don't think I mean, there are artificial ways of enhancing ciliary dyskinesia and also ciliary, you know, reducing ciliary beat frequency, but I'm not sure that they would be a way of preventing infection. In fact, I think that they would probably lead to a greater uptake, or viral uptake.
Super. Okay, so I'm going to go to a question from Vinciane Saint-Creek from INRA Paris. She thanks the speakers for excellent talks, and this question's to Oliver this time. You didn't see any effect of IL-13 on ASL volume. Did you have a look at pH? And are claudin-forming pores known to have differential permeability to bicarbonate?
Well, I think you're speculating about the change in the pH of the surface liquid according to this transport. Well, our latest results from yesterday told us that there is an alkalization of this liquid layer. So probably bicarbonate transport is somehow important for this compensation. However, we have not clear evidence concerning inhibitor that inhibits like carboanhydrase, the the bicarbonate transport, but well, it is possible, yes.
Okay, then we have a question from Pei-Ying Fong, and this is directed to Claire Smith. Is it known whether the transepithelial resistant is different in the RSV infected cells, which are neutrophil naive, so the visibly intact preparation?
Yeah, that's a really good question, because obviously, you know, we're getting a greater amount of neutrophil infiltration across the epithelium. So thinking that you would get a disruption in tight junctions would be the natural thought, but that isn't what we found. We have measured transepithelial resistance in this model, and we don't find a difference until the neutrophils are added. So before the neutrophils are added, we haven't seen a difference with RSV infection. I said that, you know, even from our electron microscopy images, we're not seeing a lot of cell loss. You know, we're just seeing a little bit of cells breaking away, but actually they're still there. So we think that the resistance is still okay. But yeah, once those neutrophils have come through, it's a different story.
Super. Okay, I'm going to go to the first question, actually, not to ignore anyone. I think, again, I'm going to direct this to Claire. Can you, it's from Timothy Danboy in Kaduna State University. Do the lymphocytes play a role in RSV infection?
So, it's a difficult question. So the lymphocytes play a role in clearing the infection. Definitely, they have a role to play in the resolution of the infection. So we're very focused on the innate immune response. So we're looking at, you know, the very early stages of infection, and I haven't come across much evidence to suggest they're playing a critical role at that very early stage. I think it's something that, you know, once you know we've got that inflammatory response in full swing, then the lymphocytes are recruited at that stage. So I think that I don't necessarily think they play an important role, yeah, at the early stage, but definitely when it comes to the resolution of infection, that they are critical in that part of it.
Okay, I would actually have a question to the two of you, just trying to combine both of your presentation, which is obviously there's obviously a big link between the barrier of the airway epithelium and then the defensive functions. And I was wondering a bit about a hen and egg problem here. So if I think of the airway epithelium as a barrier trying to be intact, controlling the water movement, the mucociliary clearance, and then based on what Oliver discussed, it senses the presence of any pathogens, including viruses. Do I understand that correctly, that basically the epithelium cries for help, opens up the tight junctions so that the neutrophils and all the immune cells can squeeze through and do their job? Or is it almost the other way around? The neutrophils squeeze through and then this feeds back on the epithelium. So what do you think is the order of events that's happening there, if it's at all possible to speculate on that?
That's a typical hen or egg question. So, well, I would say as soon as there is a situation that opens the tight junction, that does not mean that it is necessary simply due to enable immune cells to get through, because you have plenty of other lateral junction complexes that also may hinder those cells to leak through this epithelial cell layer, and probably they are also used to for the movement of these immune cells. Well, I think it is part of the story, but if it's the hen or the egg, I really don't know. Probably Claire knows.
Well, I think, I don't know if it's as black and white as that. I think that there's probably a loosening of the tight junctions in response. You know, I mean, the cells are definitely going to be releasing these cytokines very early in infection. We see it in our model that they're crying out for help. But obviously, we're not seeing a lot of damage. So I think it's probably a loosening effect but once those neutrophils have come through, that's when you've got the biggest release of the tight junctions. Because there's definitely a squeezing that's going on where they're not just falling through. And we've seen that with the receptors which are on the neutrophils which are coming through, that they are changing. It's not just a neutrophil, they don't look the same on either side of the membrane. They're changing their receptors to attach to particular proteins as they're squeezing through the epithelial cells. So yeah, I think it's probably a bit of a bit of both. You've got a loosening, and then the neutrophil is helping to actually release it entirely.
And I think eventually, sorry, Morag, just to comment on that one, you showed in one of your slides, Claire, this balance. I think this is probably what it's all about, and what determines the outcome of a disease, whether it is an infection or anything else. If something flips in either direction, this could decide between very beneficial effect and something going severely wrong, as what we observe in COVID right now.
Yeah, absolutely. I don't think if it was something that was just black and white, we would have discovered it already. So it's got to be something between the two, something that's naturally occurring but slightly shifted in the severe cases, and it's just teasing that out. And if we can find it at a level which before somebody gets infected that they're more at risk, then that's where we're going to be able to lead to this. Or even if we can discover it once they've gone infected, for this early stage of infection, that we can shift it back. So if they've just slightly gone over the edge, but we can shift it back to the good way, then we might be able to pull them back from severe disease. Definitely.
Okay, we're going to squeeze in one more question just before we finish up. So this is to Oliver. This is from Ines Cabrita from the University of Regensburg. Thank you very much to Oliver for your exciting results. Does the shortness of cilia disturb the tight junctions? And is the ciliary clearance relevant to sense TGF-1 beta? And you're on mute, sorry.
Can you repeat it? I have an awful internet connection. I couldn't understand you, sorry.
Okay, so the first question was, does the shortness of cilia disturb the tight junction? And the second is the ciliary clearance relevant to sense TGF-1 beta?
Well, the shortness of the motile cilia does not affect the tight junction integrity, I would guess. I mean, if you have the activation of this receptor and you have transport processes that involve the machinery of the motile cilia, you can expect that there is a change in the motile cilia length upon those effects. But I don't believe that this trigger has any impact onto the tight junction per se. And the other question was if the mucociliary clearance might be important for TGF-beta. Well, TGF-beta must be released from somewhere, and usually it is in a mute stage somewhere linked in the extracellular matrix. That can be the fact, and in fact the case for the mucin. And if you have a movement in the disruption of this mucin structure, maybe there is a release of TGF-beta from those complexes, but this is quite speculative. Probably these TGF-beta can be transported together with the mucin layer upwards and can cause damages somewhere else in other regions of the epithelium, other than those that are the primary site of damage and TGF-beta release.
All right, then thank you very much. I'd like to thank, first of all, again, our two speakers, Claire and Oliver, for delivering such fantastic presentations. I'd also like to thank everyone joining us this afternoon.